Hi, welcome back. This is chapter 9, Extranuclear Inheritance. I've broken this into 2 videos, but I feel like they're fairly short. This is just to separate 2 big concepts that I've put together here. Before you start, you should be confident in the definition of a nucleus, the membrane surrounding the nuclear material in eukaryotes. What is cytoplasm? The goop around the inside of the cell. Mitochondria and chloroplast, which are 2 of the organelles we talk about in eukaryotes. I want to get more specifically into their roles and the way they reproduce. And I want to ask yourself, just so you're sure, where are your genes actually located? And where do you get them from? Now, I think if you've gotten this far in genetics, you know all those answers. But just put them from the forefront of your brain, because the big ideas we're going to talk about today are the outcomes of crosses between 2 parents, how the heredity looks if that heredity is actually coming from the organelles, not from the nuclear DNA. So what about the genes that your mitochondria, or if you're a plant, a chloroplast make? And then let's look at how those codes inside of those 2 organelles express themselves, where they come from, and how they actually look a little bit more like bacteria. In the second video, we'll talk about how they got inside of the cells, and then some more horizontal gene transfer, but this time in eukaryotes, and then look at the consequences for human health and human heredity based on those things. So a little bit about organelle heredity, which sounds a little weird. I think you might be somewhat familiar with the idea that your mitochondria actually have their own DNA. In Bio 1, we cover the idea of the theory of endosymbiosis, that mitochondria and chloroplast actually arise from bacteria that were engulfed by our ancestors, in the case of mitochondria, and by the ancestors of plants for chloroplast. We've kind of been working under the assumption so far that that's not important. We've been assuming that all of your chromosomes and all of your genetic material, including everything that controls your phenotype, what you actually express, is coming from your actual DNA inside your nucleus. And we looked at in the bacteria the fact that bacteria could get extra nucleoid coming from not their inherited chromosome DNA. They could get plasmids. They could pick up from, hopefully, if our P-Glow experiment works, we will be able to observe the picking up of DNA from the surrounding environment and transformation. But in eukaryotes and sort of our ancestry, we don't do that picking up of DNA quite in the same way that the bacteria do. Now, we do have DNA that is not in our nucleus. That's all extra nuclear DNA. It's outside of your chromosomes. Most of that and most of this lecture is going to be all about the organelles that you inherited from your mom and the chromosomes that those organelles carry and how they express and expect your phenotype. I have just on this slide a mention of infectious heredity. We do know that certain infections and associations with viruses and other things can change your genetic pattern. That's outside of the scope of this lecture, but I want to bring it in here now. And if we have time to talk about it towards the end of the course, we will. And then I have a little hint at the end of lecture 2, or video 2 for this, about the maternal effect. Sometimes your phenotype is more affected by your mother's phenotype, regardless of what your genotype is. And we'll talk about that in a couple different species and look at the inheritance patterns that arise from that. That's going to cause a pattern of inheritance that we haven't seen in chromosomal or nuclear DNA expression. So when you inherit chloroplast, if you're a plant or an algae, or you have mitochondria if you're a eukaryote, these actually arise from bacteria that were moving around in the environment billions of years ago. Bacteria evolved about 3.5 billion years ago, as far as we know. The moment where a mitochondria became a mitochondria, a bacteria was engulfed by something that was ultimately going to become a eukaryote. That happened about 2.5 billion years ago. And those functionality of the mitochondria and later the chloroplast has over time, the last couple billion years, really become entwined with the eukaryote they're in. Their function and their ability to do their job right, I'm going to use the mitochondria here as an example more, the ability to do cell respiration, is intimately entwined with the nuclear DNA of the organism that it's inside of. Your mitochondria work partially because they carry their own DNA and can make their own proteins and do things that aerobic bacteria 2.5 billion years ago did on their own. But because you are in a symbiotic relationship with them, your cells also take over the role of some of the proteins they need. They actually can't be pulled out and exist on their own because they're unable to make all of the proteins they need to function as an organism. And the vice versa of that is true as well. You can't make all the proteins you need to function as an organism without your mitochondrial DNA helping you. Now, normally what we've been talking about is that the DNA that you're expressing is coming from the contribution from an egg from your mom and a sperm from your dad. Each gave you one haploid set, and your diploid, you express a combination of your mom's and your dad's genes. It turns out your mom gives you a little bit more than your dad. Your mom gives you her DNA, but also because of the nature of an egg, having a lot of cytoplasm and all of the organelles that make that egg function, your mom also gives you all of her organelles, including her mitochondria, and if you were a plant, your chloroplast, that also have their own DNA. So technically your mom gives you a little bit more DNA than your dad, because as far as we know right now, your dad really doesn't contribute substantially to the donation of organelles. I will say, if this is going to be your thing in the future, we do know that there does appear to be some transfer of mitochondrial DNA from the father, but the studies are really, really new, and that kind of will change how this works in the future. But for right now, we don't know enough about that for me to give you any pattern of heredity for it. It may just be an uncommon occurrence. Not all of the organelles in a cell are the same. And I don't just mean that like some are endoplasmic reticulum and some are mitochondria. Even your mitochondria, despite the fact that you inherit them all from your mother, actually vary in their genetic combinations. They're a little bit different than each other. We talked about in class a couple times the word hetero. Hetero means different. We talked about heterozygous, which meant that you had two different contributions in your zygote of the genes that you were going to grow up and develop with. We talked about heterogametic, having two different gametes. So whoever is the heterogametic parent passes on two different types of sex chromosomes. In XY systems, that's the male. In ZW bird systems, that's the female. Now I'm introducing another hetero word to you. Hetero means different. Plasmy means in the plasma membrane or inside the cytoplasm. So inside the plasma membrane or in the cytoplasm. In your cells, your mitochondria are heteroplasmic. They don't all have exactly the same genes. And they stem from bacteria. They weren't all identical when you got them in the first place. You got multiple mitochondria from your mom, not just one. So that leads to some of your mitochondria can do things differently than your other mitochondria. And you, I put this right on top of it. This is also true for chloroplasts in plants. I will try to go back and forth between chloroplasts and plants, but I did lean into mitochondria because we don't have the chloroplast. But I do want to highlight a little bit about chloroplast as we go forward. When we look at chloroplast expression, it's a little bit easier in some cases for us to see it with our eyes because chloroplasts are what make plants green. If you remember from your other bio class, chloroplasts are where photosynthesis actually happens. They are a thylakoid. I'm afraid another picture is going to appear here. They are a thylakoid membrane with lots of little stacks of membranes on the inside. They're covered in chlorophyll, And as the light strikes these little organelles, it converts that light and carbon dioxide that it grabs from the atmosphere into organic sugars. It traps red light and a little bit of blue light and reflects green light. So to us, chloroplasts and the pigmentation inside of them look green. That means when we see something like this, and this is based on a 1908 experiment by Correns, when you look at something like this, I can see that here in this part of the plant, the chloroplasts are functioning. They're reflecting green light and absorbing red light. Here, I can actually see that the chloroplasts are not functioning. There's either no chloroplast at all, or they're not reflecting green light. When we look at this pattern, these are 4 o'clocks. It's actually an interesting kind of flower. These 4 o'clocks seem to have a variegated pattern. And variegated means either stripy or alternating between 2 different kinds of pattern. The question became, how can you get a pattern like this? It's half green and half white, or white on some places and green in the other. This could be codominance, but we're looking at something here that really doesn't have 2 different kinds of cells. It looks like more like bar body expression to me, and something without X and Y chromosomes. So the plant is actually getting its green from the chloroplasts. If it has a chloroplast with a mutation, then what we're seeing is that chloroplast not functioning properly. Chloroplasts with mutations are white. When I have these white areas here, not to give you a huge spoiler, those chloroplasts are from an inherited line of chloroplasts that do not work properly. What we're actually seeing here is that this plant has inherited white chloroplasts and green chloroplasts from its mother, the white or variegated pattern. It turns out as we look forward, and Correns did their experiment and later experiments, it actually showed that these chloroplasts were coming because of heteroplasmy from the mom, not from the dad in any contribution. When I look at the algae Chlamydomonas, I want to show you a little bit about its reproductive pattern before we start. This reproductive pattern is similar to the one in fungus. I do not know if you have done this before. I think some of you might have done it in Bio 1. Some of you have not. So I want to have a quick segue and go over the reproductive pattern of basically fungus or many algae. If this is something you look at and you're like, oh, I totally get it, you can skip me here. This is not on the test. This is just to make the next part make sense. Chlamydomonas is a type of algae that is related to plants. It's sort of it's related to the ancestor of plants. It has both a mitochondria and a chloroplast. When they reproduce, Chlamydomonas spends most of its life as a haploid organism. So what you're looking at here, these guys, they do go through mitosis. They can become reproductively pass their genes on by going through basically cloning. But they also, even though they are haploid, they also have the opportunity to go through meiosis in a certain part of their life cycle. The way that works is we have here, it's in this part of the diagram. You have 2 different mating types. And in Chlamydomonas, there are 2. In some fungi, there are hundreds of kinds of mating types. The mating types differ often by having different alleles, different versions of genes. They recognize each other by sending chemical signals to each other, well, sort of like pheromones. They can recognize if they are the same type of mating type or different types of mating types. In Chlamydomonas, we have a positive and a negative mating type. So this is mating, MT positive is mating type positive, and MT negative is mating type negative. When these 2 different mating types come together, they can actually form a tube between them and share cytoplasm. And that results in a cell here that is basically, it has the nuclei of the positive mating type and the nuclei of the negative mating type. those nuclei will fuse, they'll go through karyogamy, and they form a zygote. This zygote here is a very short part of their lifespan, or their life cycle, that is actually diploid. You have to be diploid to go through meiosis. They form two different varieties of things together. They form a diploid cell. In this case, this diploid cell kind of travels off and away from the parent where the parents lived. It goes through meiosis and forms haploid spores or haploid babies. When it goes through meiosis, there is some crossing over between the genes of the positive and the negative mating type, but essentially they pass through a mixing up stage and then produce offspring, half of which are negative. Let me write MT negative, and half of which are MT positive. So they pass on with a little bit of genetic variation, 50-50 mating types, sort of like the XY and XX mating system, or the ZZ and the ZY, or the XO and the XX mating system. Everything we've talked about, but temperature dependence and haplodiploidy, they pass on half plus, half negative. If I drew this out in a chart, my positives and my negatives together produce half one mating type and half the other type. They are not male and female. We can't call them that because they're not producing sperm or egg. And the sort of baseline definition for when I talk about being male or female in biology is really what type of gamete you are producing. In this case, because they're producing positive and negative individuals, we call them mating type positive and negative. If you do any kind of work with fungus or algae or the protists outside of the big plants, animals, and fungus groups, these kind of mating systems are very common in the eukarya, where there's a lot of haploidiness and then a little bit of time with diploidy, or alternating through multiple stages of this in multiple hosts. This haploid single-celled eukaryote has one chloroplast. So this guy, he has one chloroplast. But in that chloroplast, there are 75 copies of a DNA molecule. So I'm going to add one gene onto the DNA molecule of the chloroplast inside the Chlamydomonas. This DNA molecule that is inside the chloroplast actually confers antibiotic resistance to streptomycin. I'm not making this example up either. When we go through the process of fertilization and I put my MT negative and my MT positive together, and we produce that diploid positive negative individual, after that moment, the chloroplast from these two original individuals also merge. So if I put this chart over here, rather than drawing on another life cycle, when I'm looking at this as a researcher, I am seeing my mating types and their streptomycin resistance. This one is with an R, resistant. With the S means susceptible. Susceptible means that they can be killed by streptomycin. When I breed them together, I would expect the same pattern that I saw in the mating types, 50% positive, 50% negative. But that is not what I see. When I put my positive and my negative together and they vary via their streptomycin resistance, I see, in this case, that they're all resistant. Now, maybe resistance is dominant. And that's what, until then, that's the conclusion I would have asked you to draw at this point, is that if all of them mix together and one of the types shows up in the F1 offspring, that suggests that that's dominance. But when I do the opposite cross with these parents, and I have my positive and my negative, but now my positive Chlamydomonas is susceptible to streptomycin, and my negative is resistant, these are opposites of each other, I find when I breed them together that in my F1 generation, I get 50-50 positive negative, and all of them are susceptible. So it's not that resistance is dominant. It's more that whatever the positive parent had is passed on, and the negative parent's contribution is irrelevant. The haploid offspring, when they come through that entire life cycle, are always like the MT plus, the mating type plus parent. The plus parent, the one with the mating type, is the one that is determining the phenotype of the offspring in that particular trait. It turns out the one that is positive is the one that is actually passing on its DNA in the chloroplasts. This is an example of a gene that's carried in a chloroplast. Chloroplasts are passed on in plants maternally, in algae and other red algae and green algae. They're passed on through the positive mating type. We also see a pattern of mitochondrial inheritance in this, and this pattern is going to be different, so turn the page in your notebook. In Chlamydomonas, we see a pattern where the positive parent passes on the chloroplast DNA, but in the pattern for mitochondria, the negative parent passes on the mitochondrial DNA. The haploid offspring look like the negative parent for mitochondrial DNA and the positive parent for chloroplast DNA. When we look at this, we're seeing a pattern of whose chloroplast and whose mitochondria, or whose organelles do the babies actually get and then use during their lifetime. This creates a really weird pattern if we tried to do a giant tree, and I'll do a phenotype expression tree in a pedigree for you as we go on. This creates a really interesting pattern of inheritance where I have to keep track of what parent has what phenotype and genotype, because the other parent's phenotype doesn't matter. One more example in this lecture, I want to talk about neurospora and yeast. Neurospora is a type of bread mold. This is it. It's called pink bread mold. This is not obvious here. You might have seen it in your real lifetime if you really forgot about something, where you'll get like a pinkish hue to your bread. You should not eat it at that point. But I'm going to give you the neurospora example. Neurospora has a mitochondrial gene, so it's inherited along with the mitochondria that slows its growth. It has some real fancy name about maternally inherited mitochondrial something something something, but everyone calls it pokey, which I really like. Oh, mitochondrial inheritance one. This is boring. We'll go with pokey. It basically impairs the mitochondria to have this gene. It's a broken gene that's not expressing the genes and the proteins that are supposed to be happening, so it slows the growth of the fungus. When I cross a wild type, so wild type means basically no pokey, and pokey, I shows that the gene is maternally inherited. You only, if you're a fungus, inherit what your mom passed on. Only one mating type actually passes it on. And I have, I want to go on to the video too, but I have in your homework an example of this. Watch as you build it. So I would build the pedigree on a separate piece of paper, even though it's on the screen, unless you can draw on your screen. Color in the parent that is actually passing on the gene. So it's a maternally inherited gene, and I think I actually did it with pokey. I had a bunch to pick from. If you follow pokey and then follow it through to the next set of parents, all the children inherit what their mother passed on to them. In animals, mitochondrial DNA is completely maternally inherited. All the children express what their mother passed on to them. Her daughters will continue to pass it on. It stops with their sons, because her sons are not passing on the mitochondrial DNA. And advice, when you get a couple layers down, you definitely want to color in that pattern rather than try to guess, because you'll get lost in the pedigree. With that, I was going to end it there because these videos are kind of short, but I want to make sure to keep theory of endosymbiosis and the explanation as the start of the second video, so you can go back to it if you need it later. I am going to, in class, talk a little bit about the tree of life, and particularly the eukaryotic tree. I have some examples for you. I've incorporated a question about the eukaryotic tree in the homework for chapter 9 and 10 as well. Don't try to worry about all of the variety on the tips of the trees. We're not worrying about what is extant or what exists now. Look at the lines that are crisscrossing the tree, showing the passing on of horizontal gene transfer, the passing on of chloroplast and mitochondria from generation to generation, where it's switching from individuals who exist at the same time. With that, I'll end it there, and I will see you in the next video. I will hopefully see you in class. We're starting official P-Glow experiments, and then ask me if you have any questions. All right. Bye-bye.